Chromatin is the complex of DNA and proteins that fills the nucleus. When the cell is not dividing, chromatin is loosely packed, allowing access to the genetic code for processes like transcription and replication. But as the cell enters division, chromatin condenses, making chromosomes visible under a microscope. This transformation is crucial for the accurate distribution of genetic material during cell division. The dynamic nature of chromatin ensures that DNA is both protected and accessible when needed, balancing stability with flexibility. The discovery of chromosomes marked a turning point in our understanding of genetics. Walter Fleming, while studying rapidly dividing salamander larvae cells, observed thread-like structures during cell division. He named this process mitosis, derived from the Greek word for thread. Fleming's observations laid the foundation for modern cell biology, revealing how genetic material is organized and passed on during cell division. His work continues to influence how we study and understand the cell cycle today. Chromatin is made up of two main components, DNA and proteins. The DNA is a long molecule composed of repeating units called nucleotides. Each nucleotide consists of a phosphate group, a pentose sugar, and a nitrogenous base. The four types of nitrogenous bases, adenine, guanine, cytosine, and thymine, pair up in specific ways, forming the rungs of the DNA ladder. These bases are joined by hydrogen bonds, creating the iconic double helix structure. The proteins, mainly histones, play a critical role in packaging and organizing the DNA within the nucleus. Histones are special proteins that help coil and package DNA into structural units called nucleosomes. Each nucleosome consists of 8 histone proteins, around which DNA winds like thread around a spool. This packaging not only compacts the DNA to fit inside the nucleus, but also plays a role in regulating gene expression. The arrangement of nucleosomes creates a repeating structure, giving chromatin its characteristic appearance under the microscope. When a cell prepares to divide, each chromosome condenses and becomes visible as two identical sister chromatids joined at a specific region called the centromere. The centromere is essential for proper chromosome alignment and separation during cell division, as it serves as the attachment point for spindle fibers. This precise structure ensures that genetic material is accurately distributed to daughter cells, maintaining the integrity of the organism's genome. Genes are specific sequences of nucleotides on a chromosome that encode the instructions for making proteins. These proteins, in turn, determine the traits and features of an organism. Genes are the fundamental units of heredity, passed from parents to offspring, and are responsible for the inheritance of specific characteristics. Each gene occupies a particular position, or locus, on a chromosome, and their expression shapes everything from eye color to susceptibility to certain diseases. The cell cycle is a series of events that cells go through as they grow and divide. It ensures that DNA is duplicated and distributed accurately to daughter cells. The cycle consists of two main phases: the non-dividing phase, called interphase, and the dividing phase, known as the M phase. Interphase itself is divided into 3 subphases: the first growth phase, the synthesis phase where DNA is replicated, and the second growth phase. This cycle is fundamental for growth, development, and maintenance of all living organisms. Interphase is the longest phase of the cell cycle, during which the cell grows, carries out normal functions, and prepares for division. It is divided into 3 stages: the first growth phase, G1, where the cell increases in size and synthesizes RNA and proteins; the synthesis phase, S, where DNA replication occurs; and the second growth phase, G2, where the cell continues to grow and produces more RNA and proteins. Although chromosomes are not visible during interphase, crucial preparations for cell division are underway. Cells divide in 2 main ways: mitosis and meiosis. Mitosis results in the production of diploid cells, which are essential for growth, repair, and replacement in multicellular organisms. Meiosis, on the other hand, produces haploid cells or gametes, which are necessary for sexual reproduction. Each type of division serves a unique purpose, ensuring the continuity and diversity of life. Mitosis is the process by which a single parent cell divides to produce two genetically identical daughter cells. This process is vital for growth, tissue repair, and asexual reproduction. The most important feature of mitosis is that the chromosome number remains the same in both the parent and daughter cells, preserving genetic stability across generations of cells. Mitosis is completed in two main steps: karyokinesis, which is the division of the nucleus, and cytokinesis, the division of the cytoplasm. Karyokinesis itself is divided into four phases: prophase, metaphase, anaphase, and telophase. Each phase is characterized by specific changes in the chromosomes and the cell's structure, ensuring the accurate distribution of genetic material. Mitosis is crucial for the life of multicellular organisms. It enables growth by increasing the number of cells, repairs damaged tissues, replaces old or dead cells, and allows for asexual reproduction in some organisms. Most importantly, mitosis ensures that each daughter cell receives the same number of chromosomes as the parent cell, maintaining genetic consistency. Meiosis is a special type of cell division that occurs in reproductive organs to produce gametes, sperm and egg cells. Unlike mitosis, meiosis reduces the chromosome number by half, ensuring that when gametes fuse during fertilization, the resulting offspring has the correct chromosome number. This reduction is essential for maintaining genetic stability across generations. Meiosis is significant for two main reasons. It halves the chromosome number in gametes, and it introduces genetic variation through the mixing of genes. This genetic shuffling occurs during the first division, when maternal and paternal chromosomes separate, and through crossing over, where segments of chromatids are exchanged. These processes ensure that each gamete is genetically unique, contributing to the diversity of offspring. During prophase, the first stage of mitosis and meiosis, several key changes occur. Centrioles begin to move apart, reaching opposite poles of the cell. Chromosomes, already duplicated as paired chromatids, become distinct and visible. The sister chromatids are attached at a small region called the centromere. Spindle fibers start to form between the centrioles, creating the achromatic spindle. Meanwhile, the nuclear membrane and nucleolus disappear, signaling the cell's readiness for division. In metaphase, chromosomes line up along the equator of the cell. Each chromosome is attached to spindle fibers by its centromere, ensuring that when the cell divides, each daughter cell will receive one copy of each chromosome. This precise alignment is critical for the equal distribution of genetic material. Anaphase is marked by the splitting of the centromere, which holds the two sister chromatids together. Once separated, the chromatids are pulled toward opposite poles of the cell by the shortening of spindle fibers. In animal cells, a furrow begins to form in the cell membrane, indicating the start of cytokinesis. This movement ensures that each new cell will have an identical set of chromosomes. During telophase, the separated sets of chromosomes reach opposite poles of the cell. Spindle fibers disappear, and the chromosomes begin to thin out, returning to their chromatin form. The nuclear membrane reforms around each set of chromosomes, and nucleoli reappear. In animal cells, the cleavage furrow deepens, preparing the cell for final separation. Cytokinesis is the final step of cell division, where the cytoplasm divides, resulting in two separate daughter cells. In animal cells, this is achieved by the deepening of the cleavage furrow, which pinches the cell into two. Each daughter cell receives an identical set of chromosomes and enough cellular machinery to function independently. Meiosis consists of two consecutive divisions, meiosis I and meiosis II. The first division reduces the chromosome number by half, while the second division separates the sister chromatids. This process results in four haploid cells, each genetically distinct from the parent cell and from each other. Meiosis is essential for sexual reproduction and genetic diversity. One of the most remarkable features of meiosis is genetic recombination, which occurs during the first division. As homologous chromosomes pair up, segments of chromatids are exchanged in a process called crossing over. This exchange creates new combinations of genes, increasing genetic variation in offspring. The X-shaped structure formed during crossing over is known as a chiasma. Meiosis ensures that gametes have half the normal chromosome number. When fertilization occurs, the fusion of two gametes restores the diploid number in the offspring. This balance is crucial for maintaining the species' chromosome number across generations, preventing doubling or loss of genetic material. It's important to distinguish between chromatin and chromosomes. Chromatin refers to the loosely packed DNA-protein complex found in the nucleus during most of the cell's life. Chromosomes are the condensed, visible forms of chromatin that appear only during cell division. This transformation is essential for the accurate segregation of genetic material. The centromere is a specialized region on the chromosome where the two sister chromatids are joined. It serves as the attachment site for spindle fibers during cell division, ensuring that each daughter cell receives one copy of each chromatid. The position of the centromere can vary, giving chromosomes their unique shapes. A nucleosome is the fundamental unit of DNA packaging in eukaryotic cells. It consists of a segment of DNA wrapped around a core of 8 histone proteins. This structure not only compacts the DNA, but also plays a role in regulating gene expression by controlling access to the genetic code. Genes are the blueprints for all proteins in the body. Each gene contains the instructions for building a specific protein, which in turn determines a particular trait or function. The sequence of nucleotides in a gene dictates the order of amino acids in a protein, linking genetic information to physical characteristics. While both mitosis and meiosis are forms of cell division, their purposes and outcomes differ. Mitosis produces two identical diploid cells for growth and repair, while meiosis creates 4 genetically unique haploid gametes for sexual reproduction. Understanding these differences is key to grasping how organisms grow and reproduce. Crossing over is a process unique to meiosis, where non-sister chromatids of homologous chromosomes exchange genetic material. This exchange occurs at points called chiasmata, resulting in new genetic combinations. Crossing over increases genetic diversity, which is vital for evolution and adaptation. In summary, chromosomes are the carriers of genetic information, organized and packaged within the nucleus as chromatin. Through the processes of mitosis and meiosis, cells grow, repair, and reproduce, ensuring the continuity and diversity of life. The intricate choreography of chromosome condensation, alignment, separation, and recombination underpins the remarkable complexity of living organisms. Understanding these processes gives us insight into the very foundation of biology.